glad science, we're glad to see you. I'm Sakshi, and today I'll be covering everything you need to know about DNA. DNA stands for deoxyribonucleic acid, and it is a self-replicating material present in nearly all living things as the main component of chromosomes. Therefore, it carries your genetic material. So let's talk about the structure of DNA. DNA is made up of nucleotides. A nucleotide consists of a 5-carbon sugar, which is called deoxyribose, and where DNA's name comes from, a phosphate molecule, and a nitrogenous base, like shown here. This would be the 5-carbon sugar, the nitrogenous bases, and the phosphate. DNA is made up of 4 different nitrogenous bases: adenine, thymine, guanine, and cytosine. Adenine binds with thymine and guanine binds with cytosine. These bonds are held together by hydrogen bonds. All of these components are organized in a double helix shape, like shown here, and it kind of is like a twisted ladder. DNA consists of 2 complementary strands running in opposite directions. You should also know that DNA replicates in a semi-conservative fashion. This means that the parent DNA will create 2 copies that contain 1 original strand and 1 new strand, like shown here. So this would be the parent, and this would be the 2 original DNA strands. So speaking of replication, let's talk about the steps of DNA replication. Replication occurs during the interphase part of the cell cycle. If you don't know what that is, you can click somewhere in the top right hand corner of your screen. The eye should be somewhere there. And so if you click on it, you will be linked to the video where I talked about the cell cycle. So anyway, DNA polymerase catalyzes the replication of new DNA. It also proofreads each new DNA strand to fix errors and minimize the chances of mutations. DNA helicase then unzips the DNA strand and breaks down the bonds that hold the 2 strands together. Then each separate strand serves as a template for a new strand. Again, A binds with T, G binds with C. So here's an example. So here, for example, A would bind with T, G with C, C with G, T with A with T, C with G, G with C, and G with C. So now that you kind of see how it's supposed to work, you can pause the video and try and do this by yourself, and then once you unpause the video, you can see the way I did it and confirm your answers. And voila, there we go. So that should be how you did it. So finally, you should know that DNA has a few nonsense nucleotide sequences at the end of the strands called telomeres. Telomeres almost act as little caps to keep the important genetic material in, because some nucleotides can be lost during the replication process. So that's all you need to know about DNA in a quick nutshell. Thank you for watching. I hope that was helpful, and I'm always glad to be of help.